Hello and welcome back. The topic for this next video are going to be proteins. All right. And in terms of proteins, proteins really have a lot of different functions within the cell. Proteins function as part of your immune system. Proteins function in making up enzymes. Proteins make up structure in the body. They move things around within the cell. And they signal or they communicate within the cell. So the cell knows where it is and what it should be doing. So proteins are really important as far as cellular structures. Proteins are going to be a polymer of amino acids. Okay, so our amino acids, that was a little squeaky, are going to be our monomer in this case. Okay, and in terms of the macromolecules, where I want you to focus right now is on proteins and their function structurally, as well as proteins and their function as enzymes. Okay. There's more than just structure and enzymes that proteins are involved in. These are the two we're going to focus on right now in this class. When you take additional classes, upper level classes, you're going to learn about some of those other functions like in the immune system, like with communication. But for now, we're going to stick with structure and enzymes. All right. Structurally, things like your tendons and ligaments are made up of proteins. Okay, so is your hair. Okay, enzymes are going to be important in chemical reactions. And in terms of enzymes, what's going to be important with enzymes, for the most part, you're going to be able to kind of pick out something that's an enzyme looking at the last three letters. So anytime you see as an ending ASE, you're talking about something that's an enzyme. In this case, lactase is the enzyme that breaks down the sugar, lactose. Okay, if you've got the OSE ending, that's indicative of something that's a sugar. All right, so this is an example how some of our macromolecules interact. The enzyme is going to be the protein. The sugar, this is our carbohydrate. Okay. So we've got a protein that will break down carbohydrates into, because if you remember lactose, when we talked about carbohydrates, lactose is one of our disaccharides. It's made up of glucose and galactose. Lactase breaks that chemical bond between glucose and galactose and gives us those individual monomers back. Okay? So lactose is a disaccharide. Specifically, the enzyme lactase breaks down lactose. All right. Where this is important in your day-to-day life, if you know anyone who's lactose intolerant, okay, they can't break down the sugar lactose. And the reason they can't break down the sugar lactose is they are lacking that enzyme. They don't have the enzyme lactase. All right. If you buy the milk, the lactate milk at the grocery store, essentially what they've done with that milk is they've added the enzyme to the milk product to break down the lactose into glucose and galactose. So if you drink side by side a glass of regular milk and a glass of the lactate milk is actually a little bit sweeter. And it's a little bit sweeter because that disaccharide has already been broken down into its 2 units. Okay? So lactose intolerance happens with people who lack the enzyme lactase. All right? Now, in terms of our structure associated with protein, because that's really what I want to focus on with this video. We talked, when we were talking about chemistry, remember I was talking about amino groups? We've got an amino group here. And we talked about carboxyl groups. We've got a carboxyl group here. Okay? So with amino acids and with proteins, this tends to be referred to as the N-terminus of the protein because you've got the nitrogen there, you've got the amino group. The C-terminus is going to be the carboxyl group. All right. Now remember, carbon makes 4 bonds, so we've got a hydrogen up here, and then what we've got down here attached to the amino acid, in your textbook that's kind of labeled as an R group. That R group is going to be some particular functional group. And I'm not so concerned in this class that you memorize the functional groups on all 20 amino acids, because in terms of that, we've got 20 different amino acids that can be used to make the proteins within our body that we need. All right, the reason we have 20 different amino acids is because of this R group. Okay, so in terms of that, this is the portion that's going to be different between each of the amino acids, so I'll go over this in green. The portion in pink is going to be the same for all amino acids. You're going to have that N-terminal, you're going to have that C-terminal end. And I'm going to draw another one over here because I'm going to show you how proteins are linked together really quickly. So we've got a hydrogen up here, and we've got an R group here as well. Okay? These might be the same R group, they might be different. Don't worry about that. Okay? And in terms of that, the R group is what gives the particular characteristic associated with the amino acid. So you're going to have some amino acids that are going to be hydrophilic, so they're going to like water, and they like water because of that R group. You're going to have some amino acids that are hydrophobic, and they're going to be hydrophobic because of that R group. Okay? You've also got some that are acids, some that are going to be polar, some that are going to be nonpolar. Don't worry about all that. R group is what varies. That's what gives us the 20 different combinations. That's what's different in that chart I've got on the PowerPoints. Don't memorize the chemical structure of all 20 amino acids. I'm not going to ask you that. Okay? But what I may ask you is how proteins are put together into longer chains. Because what we've got here, this is our monomer. Okay? We've got another monomer here. All right? And in terms of making or linking these together, the reaction that we've got there is going to be something that you're already familiar with. Because if we look right here, we've got an oxygen and we've got 2 hydrogens. Oxygen and 2 hydrogens gives us water. Okay? So linking amino acids together is another example of a dehydration synthesis. And what we get when we remove that water is we create a bond between the C-terminus of one of our monomers and the N-terminus of our next monomer. And in terms of that bond, this is going to be a covalent bond But this particular bond, covalent bond, has a special name, okay? That covalent bond, because it's linking two amino acids together, is referred to as a peptide bond, all right? So the peptide bond is a type of covalent bond. It links two amino acids together, okay? And it links the C-terminus of one amino acid to the N-terminus of the other. Now we no longer have monomers. Now we're starting to build and link together our polymer. So poly meaning many. So you link together many amino acids to make various proteins, to make our enzymes, to make the structure that makes up your hair follicle, to make the keratin, which is the protein that's involved or found in your hair follicle, okay? So in terms of that, that's what I want you to understand. I also promise I'm not going to ask you to draw an amino acid. I draw it up here so you can see where that chemical bond happens, okay? But I may ask you to recognize this structure as an amino acid. So know that that nitrogen, 2 carbons. So essentially we've got a carbon with an amino group and a carboxyl group associated with it. That makes up our amino acid, okay? But I won't ask you to draw it, I promise, okay? I do want you to know what the different components are. I want you to know the peptide bond that forms linking our monomers together, and that's going to be a dehydration synthesis, okay? Likewise, breaking a peptide bond, that's going to be a hydrolysis reaction. You're going to have to put water in to break that peptide bond, right? If you have any questions, please let me know.